So a few people have mentioned that they've been having a hard time with BioRender, where it's either not letting them log in or not letting them save. I think this wasn't an issue last year, but I think what's maybe happening is that dozens of people are on it at the same time, and I think it's kind of bogging down the account. So this is what I'm going to do. I'm going to create a second account that everyone can use. I'll start a GoFundMe if you guys also want to contribute to it. But you should be able to, if whatever you've done, if you're good with what you have, you can submit that. You can also save and export that and upload it to this other account if that makes it easier. But hopefully this will help alleviate any issues with the account being bogged down with lots of people using it. And I'll also push back the deadline till tomorrow, because I know a lot of you are probably still working on it, which is not a problem, but I'll just push back the deadline one day so that you have that time to work on it. And are there any other questions? Yes.
So with that in your illustration, I know what assignment that makes a problem and then you're proposing
it can include your methods. So for some people, they are interested in trying to do some sort of gene therapy potentially, and they could include in that illustration what that might look like. For others, it might be, it might not include that. So it doesn't have to include the method. And for some of you, you might still be figuring out what that method is. So for now, the best way to focus on what your illustration is communicating is what the problem is and what the proposed solution or improvement to the therapy or new mechanism might be. Are there any other questions? All right, we will go ahead and get started. So we left off last class, talking about different methods. And one of the methods we talked about was SDS page. And what we're going to focus on is now pivoting to Western blotting, which is a way of taking SDS page and being able to interrogate specific proteins that are present in your samples. So we talked about this a little bit last class, and to help try and illustrate the point, I actually tried to bring some equipment that might help understand a little bit more of what's actually happening. So typically we have this box that we have our gels in, and we'll actually connect it to a power supply. All right, and we'll have a certain voltage that's running across it. And so what is going to end up happening is, I mentioned before that in your gel, you'll probably have one lane that has your ladder or standard that has like specific molecular weights. It's going to be like specific proteins that mark the different molecular weights on your gel. And your samples will also have what's called a loading buffer. We didn't talk about this last class, but this is to help try and illustrate, to help you understand the picture of what's happening. So, you can't actually see the proteins, but what we do is we add this dye that's in this loading buffer that can actually help you see as your samples are running down. And sometimes you might actually not have a good connection and it's not going to run down smoothly. You might have some smiling, some frowning. It might also affect your own smiling and frowning when you're looking at your gel. But what's happening is your samples are going to be running down. You're not going to see three different colors like this. You're going to see your ladder, the separation of your ladder, and you're going to see your sample going down across your gel. And so what ends up happening next, that's really important, is that we cannot, it's really practically hard to use antibodies on a gel. If you remember, this gel is this matrix network of polyacrylamide that your proteins are embedded in. It would be incredibly hard for our antibodies to be able to diffuse through and to try and get access to these proteins. So what we do is we transfer them over to a membrane that is really amenable to us using antibodies to interrogate them. And so what we do is we'll take this cassette, we will load it up with a sponge filter paper, then the gel and your membrane. The membrane and the gel are gonna be right on top of each other. And you'll have more filter paper, more sponge. And what's happening is you are trying to create a really tight connection between your membrane and gel. And you're trying to create an environment where you're able to create across an electric field, have your proteins go across to this membrane, okay? And there's a lot of finicky parts to this entire process of Western blotting because I mentioned you can have smiling and frowning if your gel doesn't work well. What can also happen that's really annoying is if you have bubbles in between your membrane and gel, and when you're actually using antibodies and staining, all of a sudden you're just gonna have a big fat circle where you hoped your protein was gonna be. But what we're trying to do is to transfer the proteins that are in the gel across onto a membrane. And these membranes, it's usually nitrocellulose or another membrane called PVDF. But what's happening is they're able to bind to this membrane, and across the order of 30 minutes, you can have all of your protein now transferred onto this membrane. Now, when we take this membrane, we're able to label it with antibodies. In the pre-lecture, we talked about antibodies. Dr. James talked about antibodies and how they have different parts of the antibody. So what's ended up happening is for the sake of both sides of this room, I've put up antibodies on the board here. We have different parts of an antibody. We have the FAB region and the FC region. The AB means antigen binding, the C means constant. And so what ends up happening is there's like a binding pocket in the antigen binding part that binds the specific protein that you're interested in. And so you will add that protein of interest, the primary antibody, your first one. And during this process, you'll do a series of washes. And what I forgot to mention is blocking, forgive me. You cannot forget blocking. So blocking is really important because you don't want to have non-specific binding of your antibody to some other protein. Proteins can naturally kind of be sticky to one another at times. And so what you want to do is you want to have some sort of abundant protein that will occupy all of the non-specific binding that can occur. We'll oftentimes pick a protein that is not subject to creating large dimers or multimers. And so we'll pick something like bovine serine albumin. Sometimes people use fish gelatin. Some people use dry milk. Some people use casein, which is a protein in milk. And so it oftentimes is dependent on what you're probing for. And so you'll block the non-specific binding sites, and then you'll introduce your primary antibody. What ends up happening next is you'll have a secondary antibody that binds to the first. And so usually what we'll have is a protein, a primary antibody that maybe is recognizing actin. So an antibody that's raised against actin, that is a rabbit antibody. Your secondary protein will recognize rabbit antibodies, period. And it will bind to the FC domain. So if you can imagine, and we'll have an illustration of this in a moment, you'll have a blot here. You'll have your antibody that binds here because the antigen binding site is recognizing a specific protein. And your secondary antibody will bind to the FC domain. Their antibody that'll bind here. What's usually nice is that the secondary antibody is usually labeled with some sort of fluorophore. And you'll be able to image the blot and you'll be able to see really nice bands that maybe changes across your different samples. Are there any questions so far? Yes. That's a very good question. I'm going to answer that in just a moment. Hold on to that thought. The question was, for the sake of recording, why do we use a labeled secondary instead of a labeled primary antibody? Any other questions? This is illustrating all the things I just talked about, forgive me. Okay, so previously we talked about Kamasi blue being able to stain specific total protein. It's not looking at anything specific, just what's the total protein content in your sample in that well, or in that lane, forgive me. Western blotting, you're able to use specific antibodies that look at specific things. So for example, in this sample, what ended up happening was this was bacteria that were introduced with a specific plasmid vector to express either ATPase or thyroidoxin. So when you're looking at Kamasi blue staining, if you see a big thick band at what your bacteria is supposed to be expressing, that's great. This is like the ideal Kamasi blue staining if you have a bacteria that's supposed to be expressing a certain protein. But Western blotting, what we're able to do is take the sample, run it, and use antibodies to detect those specific items. And so what we're able to see is that the entire bacterial lysate has a huge expression of thyroidoxin, and in a purified form, this is what it looks like. And for a separate bacteria that was raised to express ATPase, you have your specific expression that is what you'd expect to see. And in this case, what ended up happening was, what's really noteworthy is that ATPase is also expressed in this sample here as well. This is the entire bacterial lysate here, as opposed to the purified thyroidoxin sample. Does anyone have any idea why they think that might happen? Why is that ATPase being detected in that sample? Yes. That's one possible explanation. There is also a simpler one. There can also be a bacteria, that it kind of ties to what you're saying. Bacteria can also express, there can be a bacterial ATPase that this ATPase antibody is cross-reactive to. And so in a case where there's the entire bacterial lysate run, there's also this ATPase that the bacteria express that also binds to the antibody. But so like this is meant to illustrate the improvement of kind of questions you can answer with Western blotting. With Kamasi blue, with Kamasi blue, you can only say, this is what the total protein looks like. Whereas with Western blotting, using antibodies, you can interrogate specific proteins and their expression. So to revisit antibodies, antibodies are a form of, you can think of them as a receptor that's against a specific foreign material. We'll talk more about this once we get to the immune system. Those lectures, I hope you guys are as hyped as I am about those lectures. Those will be great. But typically the foreign material that an antibody binds to, we call that the antigen. And so antibodies are typically meant to recognize specific antigens. And so in Western blotting, what we do is primary antibodies bind only to the antigen or protein of interest. And for secondary antibodies, they bind the antigen, for them in this case, is the Fc portion of the primary antibody. So for example, the primary antibody that we used was raised in rabbits. And so it's a rabbit antibody, and it recognizes the catalytic subunit of pKa. Typically in antibody nomenclature, we say rabbit for the species that it was raised in, anti for what it recognizes. So rabbit anti pKaC. The secondary antibody, we'd want it to recognize that a rabbit antibody, those species that it came up in. We'd want to use a completely different species. We wouldn't want to use a rabbit anti-rabbit, because then that secondary antibody can be binding to itself. So what we do is we have a goat anti-rabbit IgG. IgG is the kind of antibodies. There's 5 different flavors of antibodies, but I don't want to scoop myself for the adaptive immunity lecture, so you can remain on the edge of your seats for the different flavors of antibodies there. But this is typically going to be either like a fluorophore conjugated, or in this case, enzyme conjugated goat anti-rabbit, because it's recognizing this the class, the rabbit primary antibody, because it's binding to the constant portion of that antibody. All right. And as an example, thinking towards the monoclonal antibodies that are raised for the sake of coronavirus treatment or for the vaccine development. So there's an important thing to note about monoclonal versus polyclonal antibodies. and it refers to how many different parts of the protein of interest it recognizes. Does anybody have any guess how many sites of a protein of interest does a monoclonal antibody bind to? One. Polyclonal, as the name implies, binds to many parts of it. And so the way we raise polyclonal antibodies is typically that protein of interest is you'll actually immunize an animal, either a mouse, rabbit, donkey, goat. Some people will use llamas. You can use alpacas. You can use dogs. But typically in biomedical research, rats, rabbits, and goats are really popular animals to use this for. And so you'll immunize the animal with it. And when you collect the sera to isolate and purify the antibodies, it could have recognized different parts of that protein. So that's how polyclonal antibodies are made, because it recognizes there are different antibodies that recognize different parts of that protein. For monoclonal antibodies, it's a totally different ballgame that takes a much longer time. And we're actually going to look at an order form later on to see what goes into that. But typically, what ends up happening is you'll have an animal that's been immunized with a specific peptide or protein of interest, and then you'll collect the B cells, and you'll actually take them and fuse them with a tumor B cell, and then all of a sudden that's what's called a hybridoma, and there are specific hybridomas that will respond to different, that have specific clones of antibodies, and you'll see which ones using ELISA what peptides they bind to and recognize, and you'll generate antibodies from those specific single cell clones, and that's how we raise monoclonal antibodies. So in the case of the spike protein from coronavirus, if an animal is immunized with spike protein, it's pre-fusion complex, it could be binding to any parts of the protein. And if we use a polyclonal antibody for spike protein, what's really important is that for spike protein, there's a pre-fusion complex before it binds to ACE2 in our cells and has viral entry, and a post-fusion. And so what we really want to focus on is in this receptor binding domain, that's the site that we're really interested in to develop antibodies for, because this is the site that binds to our cells, and this is the site that changes from the pre-fusion to post-fusion complex. And so, for example, we'd want to raise monoclonal antibodies against this part of the spike protein. And so, for example, like the Regeneron monoclonal antibody treatment, it's actually made recognizing that part of the pre-fusion spike protein. So that's an example where having a monoclonal antibody is really important, and there might be questions for Western blotting where monoclonal antibodies are needed. Yes?
Like primarily through like right, like sure, but like can we also be like synthetically produced?
Yes, so it's a great question. That's actually something that our lab does, or like one arm of our lab's work. You can synthetically derive antibodies, and not only that, but you can also change how much of the antibody is of what you want. So like, you know, an antibody is really large, and if you want it to cross into something, it's really hard for an entire antibody to do that. So instead, what you can do is you can just have this tiny Fab region that becomes a single chain fragment. And there are a lot of therapies that are in the pipeline that are just like a single chain fragment of an antibody that has the antigen binding site, but is much smaller, can cross into spaces that an entire antibody can't cross. And there's also a technology called phage display where you'll use bacteria to just generate a bunch of different peptides and create antibodies that recognize specific peptides that you're interested in. And so like that, I'm pretty sure won the Nobel Prize within the last 10 years. And a lot of antibody treatments coming out of pharmaceutical companies are like taking like little chunks of antibodies and using them that way. That was a great question. Yes. You're stealing my answer for her question, but you got to a lot of it. Yeah. So typically, we don't always use monoclonal antibodies for our primary. It depends on what question you're asking, but a lot of times that is the case. and they're really hard to generate and really expensive. And so it's tagging them with a fluorophore can be inefficient. And so because of that, we'll use secondary antibodies that can amplify the signal a ton. Usually it's not just one secondary antibody binding here. These are polyclonal, and so they're binding to a bunch of different parts of the FC region. And so like one primary can have lots of secondary on it, and that amplifies the signal a lot. I was trying to save that answer for later, but you basically said it. You're good. Are there any other questions? These are great questions, by the way. All right. So why use secondary antibodies? So the secondary antibody is directed against, like I said, the FC domain of the primary antibody. And so regardless of the specificity, the secondary antibodies can be mass produced because they're polyclonal. They're not meant to be binding to a specific part of the FC domain of the primary antibody, but rather can be mass produced and are easily made because they're polyclonal as opposed to monoclonal. The secondary antibody, this is going to be important, can be wastefully coupled to a chromogenic enzyme. Chromogenic means like making a color. And so we'll talk a little bit more about that in a moment, but they can be wastefully coupled to that chromogenic enzyme or a fluorophore. And it's wastefully because it's not oftentimes an efficient reaction. And so for doing that reaction with a secondary antibody is a lot easier because, like to the first point points to, they can be mass produced. Whereas if we're doing that with a really special important primary antibody that's monoclonal, if we're having efficient tethering of that enzyme or a fluorophore, it's not going to be great. It's going to be it's going to be wasting a lot of money, and it's going to lead to results that can be hard to interpret, not be representative of what's actually happening. So instead, we use secondary antibodies that can amplify the signal and lead to easier detection. And so what ends up happening is multiple secondary antibodies will bind to each primary antibody, and this amplifies the signal. So like this is a crude illustration of what happens with a Western blot, where we have our membrane with all of our proteins on it, and our protein is at this specific site where our primary antibody is bound to it. But then we'll have many secondary antibodies binding to it on the FC region, and each secondary has either a fluorophore or an enzyme on it. Typically, the enzyme is horseradish peroxidase. And like we learned in our enzyme lecture, every enzyme has a specific substrate that it's targeting. And so you'll actually add something that's typically called substrate, and that'll lead to the color change that you can then use to actually image and get a sense of how is your protein there. And so typically, primary antibodies, you'll have very little of it. Speaking from personal experience, it's always funny trying to order a primary antibody. They're oftentimes way more expensive than you think, on the order of like $300 to $500 for what equates to like 5 droplets of it. And so, the more special that antibody is, the more expensive that is. Like 300 to 500 is usually the average. And so you have to, so that's why for primary antibodies, we're usually trying to be very conservative with how we use it, but then leverage what we can do with secondary antibodies to amplify the signal and not be wasteful. I'll wait for a couple of questions there. Yes. Yeah, so in that example, the receptor binding domain of spike protein, it usually exists like it's a trimer, but for spike protein, it has its pre-fusion complex and the post-fusion when it binds to the ACE2 receptor. And, one of the antibodies typically have lots of different functions. They can bind to a cell based on its FC region, and different antibodies will elicit different responses. But also, the antibodies can bind to something with its antigen, with its FAB site, and neutralize whatever it is. And so, one way that monoclonal antibodies can work in the case of coronavirus is that they can bind to the receptor binding domain of the spike protein and its pre-fusion complex. Post-fusion, that site's not really accessible because it's bound and beginning viral entry into our cells. You can bind and neutralize the receptor binding domain site, and that gives time for your immune cells to locate and destroy that virus. Also, when it's bound, the FC region is sticking out when it's bound to the coronavirus. And so there are cells that will recognize that FC portion and say like, oh, there's something here that the antibody is bound to, we need to destroy it. And so being able to bind to specific sites like that can be really important for immune responses. For biochemistry techniques, there can be questions about specific sites that are phosphorylated. And this might seem like this is one of those like basic science questions, but oftentimes can lead to big downstream drug therapeutic implications, where specific sites that if they're tyrosine phosphorylated, or if a specific protein phosphorylated here or serine phosphorylated here, could that lead to different outcomes? And what for some cancer drugs that have been manufactured, being able to look at specific tyrosine phosphorylation events have been really critical to drug development. And so being able to interrogate specific sites that are phosphorylated at specific areas can be also really important. That's a great question. Does that make sense? Okay, good. Are there any other questions? All right. So this is an order sheet that the Janes lab used to develop a specific monoclonal antibody for serine phosphorylation for a protein of interest that they have. So typically what ends up happening is when you raise these antibodies against a specific peptide or a specific protein, you also need to have that peptide to do a confirmation study to figure out, does it actually bind to what we have? And so what they did is they had a specific protein where they used this company to make this specific peptide. They have the phosphorylated and unphosphorylated part of it. And what they ended up doing was using rabbits to raise this antibody. So peptide was injected into rabbits. And you can kind of like see the price tags with each of these items as it collects. But what ends up happening is the really hard part is then the antibody purification part, to try and get a sense of which antibody has the highest affinity for a specific peptide. And so the company raised, I refer to these hybridomas. So like what ends up happening is in these rabbits, the B cells, those are the antibody producing cells in our body, B cells are harvested. B cells will be fused with what's called a myeloma, these cancerous B cells basically, and they're fused to make a hybridoma. Those are the cells that, what ends up happening, the benefit of that is you have, when they're fused together, you have the antibody production part of the B cell, and the cancerous part of it means that it can keep going and keep living, and it'll keep proliferating. So you have these different hybridomas that are each of them is a different clone. So this goes to the monoclonal, polyclonal. Each of them will make a different antibody that recognizes a specific antigen or a specific part of the antigen. So for example, whatever protein this antibody is binding to, that's the antigen. But the different parts of it that it recognizes are called epitopes. So monoclonal antibodies recognize specific epitopes on an antigen. I'm going to say that again slowly so that makes sense. So the protein of interest is the antigen. The different parts of it are called, that antibodies recognize are called epitopes. Monoclonal antibodies will recognize specific epitopes on an antigen. And so what they're trying to do here is find the clone of the hybridoma that has the highest affinity for binding the epitope of interest here, which is their peptide. All right. And so over time, this gets up to be about $5,300. That's fairly inexpensive. So the one in our lab that's being developed to get 10 micrograms, which is an amount you can barely see, that's between $30,000 and $40,000. So like this can become a really expensive endeavor, but will either likely answer a really important question for their sake, probably in cancer biology, or for developing some sort of therapeutic in fibrosis, like what our lab is trying to do. And so the benefit again of monoclonal antibodies is that you can bind specific epitopes that are really important in either answering a question or finding a new drug. Are there any questions there? So for example, if I go back to the spike protein example, the entire spike protein, is that an epitope or an antigen? Antigen. The receptor binding domain that monoclonal antibodies that are used clinically, if they're recognizing the receptor binding domain, is the receptor binding domain an epitope or an antigen? Yes. And so typically the epitopes are going to be short peptide sequences. And when we get to that, I'll try and have an exercise that helps to illustrate that. I think it's going to be fun. If nothing else, it's going to lead to a snack that you guys can enjoy. But the ability for monoclonal antibodies to bind specific epitopes is really important. And so when you're raising up antibodies in animals like this, typically what happens is it's about a 70 to 80 day process. So typically what happens is first the animal is, this is going to sound disturbing, I apologize, they're pre-bled, which means you get a sample of blood to look at, is this animal already undergoing strong immune responses that is elevating their antibody levels systemically. Oftentimes you'll pay a little extra for them to be in pathogen-free environments, so that typically they should not be having undergoing a severe or strong immune response that will kind of mess with the antibody levels if you're trying to collect antibodies from these animals. So assuming everything's all clear, you'll do what's called the primary subcutaneous injection of whatever it is you want to raise the antibodies against for. And it'll be 500 micrograms with what's called Freund's complete adjuvant. This is how antibodies used to be done. This is how vaccines used to be developed for people. It's not the case anymore. But Freund's complete adjuvant, an adjuvant, does anyone have an idea what an adjuvant means or what that is? Does that sound familiar at all? So adjuvants are usually things that help raise the immune response that are in a vaccine, or in this case, what are antibodies being raised against. And so Freund's complete adjuvant, they're mineral oil, surfactant, and dead mycobacteria. So the mineral oil is meant to help elevate the viscosity of what you're injecting, so that when you inject it, it doesn't just diffuse and go everywhere, but rather it stays together. The surfactant is similar to what it does in the lung. It reduces surface tension, so that when it gets to, it's able to get to its intended site and travel through the aqueous environment. And finally, the dead mycobacteria. This is basically taking a sledgehammer to your immune response and whacking it on the head to wake up and drive your immune response to become activated. We do not develop vaccines anymore that have complete adjuvant. For example, the coronavirus vaccine from Pfizer and Moderna, what's so special about them is that not only do they lack mycobacteria, but they lack some of these other adjuvants and have much simpler components that help with entering cell plasma membrane. So as you can imagine, they have lipids that help them cross across the plasma membrane, and then salts and sugars that help with other interactions. But our vaccines that we're developing now do not have this. And for animals, more recently, they're not using complete adjuvant because this also leads to some necrosis and toxicity in the animal. But this is just meant to help historically illustrate how we developed antibodies. And so you have your first primary subcutaneous delivery with Freud's complete adjuvant. A few weeks later, they'll get a booster. So with the first delivery, the immune response is primed to this specific antigen. If you can imagine, I oftentimes anthropomorphize the immune response, that they've seen this antigen, they've seen this thing, they have T cells that recognize it and now B cells that recognize it, and they'll say, okay, we know what this is, okay, maybe not maybe, but we'll remember it. But it's not a very efficient immune response. The affinity of those antibodies is low. The affinity of the T cell receptor is low. So what ends up happening is you boost it to improve that immune response, improve the affinity of the antibodies, and improve the response of the T cells. And so what ends up happening is you do that with Freud's incomplete adjuvant, which means it has no mycobacteria. So you don't need to whack it over the head any longer to trigger that immune response. But now what you're doing is you're improving the affinity and improving the response of the immune cells to this specific antigen. And so you'll do another booster at 42 days. And then after that, what you'll do is you'll do another bleed to look at and trying to identify, do you have any antibodies in your serum that recognize your specific antigen of interest? And you'll do an experiment where you'll use a peptide to try and see if the antibody recognizes it. And in the case where it's necessary, you'll do another booster of subcutaneous injection of your antigen, and you'll do another bleed, and you'll euthanize the animal to collect as much blood as possible, to collect as much antibody as possible. And so the way in which we test whether these antibodies are recognizing the antigen of interest is something called an ELISA, an enzyme-linked immunosorbent assay. And there's different flavors of ELISAs. So this is going to be a direct ELISA. The way this is going to work is, for a direct ELISA, you're taking your antigen of interest and you are coating the bottom of the plate with this antigen. These plates, we call them ELISA plates because they've been modified. It's typically, there will be some passive absorption of a protein on the bottom of a well for an ELISA. But what ends up happening that's a lot better is that these will be treated, the bottom of the ELISA plate will be treated so that it has a charge, and that when the antigen is being coated, it'll be in a coating buffer that has a really high pH. And so that will create electrostatic interactions for your antigen to be adhered to the bottom of your plate. And what's important is that with each step that we have here, we're going to be washing the heck out of the well. And these interactions are so good in that one of the ways that we discovered we could wash these wells really easily is just dunk them in a huge bucket and pull it up and just pat it dry, instead of just like pipetting in each individual well or with a multi-channel. So these interactions are so good that you can just dunk it in some sort of solution to wash it and dump it out. And next, again, blocking is your friend. You want to reduce as much non-specific binding as possible. So we use a really abundant protein that'll occupy all of the non-specific binding sites. Because we're using antibodies here that are specific, we want to get rid of all of the non-specific binding. So whatever you use here, oftentimes it can be albumin, as I mentioned before, it can be casein or milk protein. You are blocking all the non-specific binding that can occur. Because again, these plates are charged and they will want to bind to proteins, and proteins can be, because we're using a charged plate, we want to try and occupy all of those binding sites non-specifically. And then now, because we have our antigen that's on the bottom, we're going to add our primary antibody. For antibody production, to see the affinity of it, this will be the antibodies that you're isolating themselves. Again, we'll wash the heck out of it. And in this case, you'll add a conjugated secondary antibody that has an enzyme with it. That enzyme, as I mentioned before, is oftentimes HRP. Yes. So imagine there is, so what we want to try and do is eliminate as much of the unbound, conjugated materials. And so, for example, when we're adding, so this step is actually a good example. So we just added our enzyme-linked secondary antibody. As I mentioned before, these wells are treated and will want to bind to proteins. Now, when we coated this plate with the antigen of interest, it was in a coating buffer that jacked up the pH that induced a bit of a negative charge on the protein. So the proteins are negatively charged. The plate is positively charged. So those electrostatic interactions are stronger than, say, if proteins are just absorbed based on hydrophobic interactions. So we can wash away, based on those hydrophobic interactions, anything that's bound that way. Because what's going to end up happening next is we're going to add our substrate in the next step. If we have secondary antibody that's kind of like absorbed to the side of the plate, that's going to tell us we're going to see more color change and making us think that there's more antigen being bound to than there is. And so we want to try and make sure that anything, even though we blocked, we want to get rid of anything that's bound that shouldn't be. And washing, and that's oftentimes with something called PBS that is pH of 7.4 to be physiologically relevant, but also has detergents that can also disrupt those hydrophobic interactions. So that wash with detergent will help get rid of anything that's not actually bound. Does that help? So we'll be doing this at each step. And after our enzyme-linked secondary, we will add the substrate, the chromogenic substrate, where you'll get color change. And you can use a plate reader that'll tell you how blue your well is. The more blue it is, the more antibody binding to antigen there is. Are there any questions so far? Yes.
What did you say that the common enzyme was that the secondary was?
It's oftentimes HRP, horseradish peroxidase. That's oftentimes used just because it's the substrate for it is really easy to use and it's easily conjugated to these antibodies. Funny enough, that's not what first things that you do. One of the first things I learned as a grad student was doing ELISAs, because it's kind of a way to kind of like get you into cranking out as many things as possible. And so when we did it, oftentimes the secondary was conjugated to something called biotin, and HRP was conjugated to something called avidin. Biotin and avidin have the strongest affinity known to nature, and so a really strong affinity to try and avoid perhaps the inefficiency of HRP with this antibody. But typically, all that's happening is the enzyme here is interacting with the substrate. You're just filling the well with its liquid substrate, and it's, as an enzyme, it's catalyzing a reaction to change color. And we are seeing the bluification of the well and using a plate reader to see how blue each well is as a way to indicate how much antibody is binding to antigen. Bluification was the really silly term that we use in the lab when doing ELISAs. Because when you're doing 10 of them at a time, you need something to help you keep your sanity. And so you use the plate reader to measure the absorbance at that specific wavelength. Now, sandwich ELISAs are really widely used. And so what ends up happening, it's slightly different, but a lot of the same concepts. Instead of coating the plate with antigen, you're coating the plate with a capture antibody. All right. Now, again, with each step you're going to be washing, you're going to introduce your block to reduce non-specific binding. Now you're going to add your antigen. So what happened before was you were coating with a specific antigen, like a specific pure solution of that specific antigen. Now what can end up happening is you can have a blood sample, a urine sample, you can have a sample from cells. It'll allow for a mixture of your sample with different proteins in it that can now, these antibodies that were coated, the capture antibody, to a specific protein in that solution. And so that specific antibody, the specific antigen is bound to the antibody, we'll wash, and now we'll add a detection antibody. So this part is really important. So the detection antibody, that the first thing that was added, those will typically be a monoclonal antibody that binds to a specific part of the antigen. Detection antibody, that will be a polyclonal, that means bind to any part of it. Because if you have two monoclonal antibodies for your detection, they're going to be trying to bind to the same part, and that's not really going to work. And so you're going to have a monoclonal antibody that binds to a specific part here, and for the detection antibody that comes in, it's going to bind to a separate part, and it's going to be a little more promiscuous because it's polyclonal. We call this a sandwich ELISA because it's two antibodies sandwiched over the antigen. And so here we'll have an enzyme-linked secondary antibody. We'll add our liquid chromogenic substrate, and we'll get color change, and we'll use a plate reader to measure absorbance. Are there any questions there? Yes. Yeah, so for example, if you're testing blood samples, if you're testing some sort of sample that has a mixture of proteins in it, you'll want to use a sandwich ELISA. So for example, typically whenever I was looking at immune cells to see how much of a protein are they releasing in their environment, I'll just collect the environment they're in, that media, and run ELISAs on them. And so that is a way in which sandwich ELISAs provide a specific answer to a sample that has a mixture of stuff in it. For the direct ELISA, for example, if we created an antibody in our lab and we wanted to know its affinity and how good it was at binding our antigen, we can run a direct ELISA where the antigen of interest, that peptide, is coded, and we'll test our antibody to see how good it is at binding. And so it's not so much a pros and cons of direct ELISA versus sandwich ELISA, but rather what's the question that you're trying to answer that might help you that might help you address whatever the issue is or answer that question. So in the spirit of what I said last class, this is a methods-heavy lecture looking at a few different techniques. So to take a step back, looking at these two different ELISA methods we discussed, the big picture here that I want you all to think about is this is a technique that leverages antibodies, whether the antibody is directly binding to an antigen that's on the plate or antigen that's sandwiched between antibodies, but allows you to try and detect and measure how much either the antigen is there in the sample or how good your antibody might be binding to that antigen. So rather than trying to focus on memorizing each of these steps, which steps you wash at, if you actually start doing them, that is important. But for the context of this class, to take a step back and to think about what is actually happening here, not on a step-by-step basis, but rather a big picture. We're having antibodies that are binding to specific antigens, and one that might help us potentially see how well an antibody might be binding to an antigen, or one that helps us detect perhaps maybe a mixture of sample that has different proteins in there to detect that specific protein. So these are all different methods for protein detection. And there are different ways to manipulate them where you might be able to detect things that cells are secreting. And this is kind of going to the projects that you guys are working on, when the next steps will be kind of considering methods that you might want to employ. If we're measuring things that cells are secreting, things that are within a cell, we talked about how there are antibodies, phosphorylated proteins. You can cell and look at how much of a phosphorylated protein is in the cell using the ELISA. But again, to take a step back and just think about the big picture and what each of these techniques permit you to do. Any questions there? All right. So why not ELISAs all the time? If you think about the ELISA versus the Western blot, in case it's not clear, ELISAs are way easier. Western blots have a lot more room for error. whether it's having a bubble between your membrane and your gel when you're transferring, or having a messed up gel when it's running, or touching your gel with your finger and messing up your gel period. There are ways that you can ruin a Western blot, whereas ELISAs are a little more simple, but there are reasons why we can't do ELISAs all the time. We're able to do ELISAs to measure, they're typically used for secreted proteins, but you can use them for proteins that are in a cell. One thing that's really interesting is this is something that the Jane's lab discovered. When we're using antibodies that look at phosphorylated ERK, that's a protein that's a MAP kinase. And it comes in different isoforms, 1, 2, and 5. The antibody that we have that recognizes phosphorylated ERK 1 and 2, if we, you can run it on an ELISA, but what ends up happening is if you take that same antibody and run it, it actually binds to ERK5 as well. And ERK is one of those proteins that's kind of like at the center of everything. And so being able to properly detect it is important. And so in the context of Coxsackie virus infection, which is some of the work that the Jane's lab does, what they were able to discover is that it's actually a mixture of ERK 1 and 2 and 5, despite an antibody that's only meant to be for ERK 1 and 2. And so what Western blotting allows you to do is it allows you to separate out those proteins because ERK5 has a different molecular weight. And so what ends up with the important takeaway here is that ELISAs, while allowing you to detect certain proteins, there are potentially shortfalls or pitfalls of our antibodies that don't do what we think they do. They're not perfect, and a Western blot will allow you to separate out those proteins, look at across their different molecular weights, and assess differences across potentially different isoforms of that same protein. And so what they're able to show is that using inhibitors, again, similar to the proteins, perhaps they might not be doing what we think they are. Chemical inhibitors also sometimes don't do what they think we are, or what we think they're doing, and they can have off-target effects, which is why whenever you see drug commercial sort of inhibitor, there's a laundry list of side effects that can also happen. But they're able to show that using ERK2 inhibitor versus an ERK125 inhibitor in the presence of that Coxsackie virus, that ERK1 and 2 inhibition of ERK signaling, the ERK1/2 only accounts of about 25% of the phospho-ERK signal, but ERK5, when you have the ERK1/2/5 inhibitor, accounts for about 50% of the phosphorylated ERK there. And so using Western blotting can also help address the pitfalls of ELISAs, where sometimes the antibodies might not be doing what they think they're doing. Yes?
Trying to get a better handle on that. So like, we don't want to use ELISAs all the time because the antibodies potentially
Yeah, so like this is one case where like that isomer, there's an issue there. So typically for, that's typically why for secreted proteins that don't have a lot of isomers, ELISAs are super easy to run and you can crank them out really quickly. When you're running ELISAs for proteins like this that have different isomers and can be quickly phosphorylated on and off. So like this is hours post-infection. It is not as easy to try and do this with an ELISA as it is with a Western blot. And so that's more of a practical, like actually running and doing the experiment pitfall. But the main takeaway was what we were addressing, that it's largely the antibodies for different isomers here, and secreted proteins usually don't have the same different isomer issues at times that these intracellular proteins do. And that's why, to simplify it, usually I always like to think I want to run an ELISA on a secreted protein, and I want to run a Western on an intracellular protein. And that allows me to separate things out and interrogate them a little differently. That's not always the case, because here Jane's lab uses ELISAs for intracellular proteins, but you just run into a lot of pitfall. You run into a lot of issues when you're trying to do it with an ELISA. There might be other scientists that totally disagree with me. Taylor could be one of them. We could have a fight about this afterwards. But that's just one school of thought. Are there any other questions? All right. So like Westerns, like the sandwich ELISA, like affinity chromatography from your pre-lecture, lateral flow immunochromatography is pretty similar to a lot of these. And so this is taking it back to the tests for a pregnancy test, an anthrax test, or the rapid COVID antigen test. And so typically in lateral flow chromatography, you have some sort of biosample. And you'll add that biosample into the sample port. What will typically happen fairly quickly is that the sample port introduces the sample into this resin. You can kind of think of that as like the polycrylomide gel for a Western blot. And so it's within this resin where there are gold conjugated antibodies that recognize specific antigens that are in that biosample. And so as the name implies, it'll flow laterally in this resin where there are antibodies coded at the test site for antigen-bound antibodies with the gold particle. And at the control site, there's antigen coded on that site, whereas the antibody gold particle coded antibodies will bind here. And so as you can imagine, the control site is meant as a proof to show that the test worked. And what will end up happening, forgive me, you'll have a chromogenic color change, chromogenic color change at the test site if you have biosample there that's bound, and as well as at the control site. We have a few folks in our community that have developed different biochemical methods. One of them is my advisor, Tom Barker, who is developing antibodies for diagnostics and therapeutics, kind of like an emerging field called theranostics. James Anders in chemistry uses capillary electrophoresis, and Kevin Janes uses a variety of biochemical and genetic methods that leverages some of these different tools. And so this was a method-heavy couple of lectures, but again, I encourage you to take a step back and try and look at the big picture first, and then try and populate that big picture with some of the finer details. With that, I was going to ask, are there any questions? Yes. Yeah, so direct ELISAs are great for detecting antibody affinity, and so typically you might also have some sort of standard that shows like color change as it relates to the affinity of that specific antibody. But direct ELISAs are great at binding to a specific antigen of interest and being able to see the color change there. The antigen will be the antigen coded at the bottom of the direct ELISA plate is going to be, for example, in that order sheet that I showed from the Janes lab, they had like a specific peptide of interest that's phosphorylated there. They just coat the bottom of the well with that thing, and then use their antibodies they made to see how well does it bind to it, and maybe perhaps there are different clones that they're raising. Which clone binds to this best? And the clone that has the best color change, that's the one they're going to run with. Does that make sense? Are there any other questions? All right, that concludes today's lecture. I'll be hanging back if you guys have any questions.